The brain is a series of connections. These, correctly structured in the right order, allow us to store memories and learn. These connections are like the tube map, where people are individual bits of information. Information travels along a track called a neuron. Each station is a point where information is processed, going forward to the next stop, changing route to go somewhere on a new line, or getting off and stopping altogether. Synapses are the stations in the brain where this happens.
When you learn something in memory, you make connections between different neurons, and those connections are specialized regions called synapses. And at the synapse, you actually have proteins that are involved in this transmission of a signal from one neuron to another.
Synapses are small gaps. They're the site where chemical messages, called neurotransmitters, are passed. These can interact with different points on the opposite neuron to bring about an effect. These sites are called receptors. They are often points of entry to the neuron called channels. So how exactly does information pass across a synapse? An electrical signal coming along a presynaptic neuron causes different types of neurotransmitters to be released into the gap. Different neurotransmitters work on specific proteins called receptors on the postsynaptic neuron. To respond to the signal appropriately, the receptor recruits a series of proteins that have different functions in the pathway.
Postsynaptic density is a specialized region in neurons that's involved in the transmission of a signal from one neuron to another neuron. It's specialized in the sense that it has lots of proteins located in there. And that's where its names come from, means it's actually a lot of proteins, very dense with proteins.
It is the ordering of the recruited protein components that allow signals to be interpreted and correctly executed. Like dispatch whistleblowers and train drivers, they allow things to run smoothly, but they must be able to interact with one another in order to pass a message along. If these proteins are not correctly structured, they can struggle to interact correctly, and so messages can be lost or misread, resulting in incorrect action.
PDZ domain is a small protein, about 100 amino acids long. It's a nice small compact protein, and what it does is involves in interactions of other proteins. So it actually binds the very C terminus of certain proteins. So it's the very last 4 amino acids that PDZ domains bind to. So they're a protein-protein interaction domain. It may sound very simple that what these proteins do is hold things together. So it gives the impression it's like Velcro, the PDZ domains. But they're really important from the point of view of memory, because they're the proteins that keep everything together so that when you do learn and that connection between neurons is made, it becomes fixed. So it's known that if these connecting proteins like the PDZ domains are lost, your memory does begin to fail. The proteins that I'm interested in are the NMDA receptors, which are involved in the signal transmission from one neuron to another, and the PSD95, which is a protein-protein interaction domains that link up the NMDA receptors with other proteins that are involved in this regulation. The way we're approaching it is once we understand the structure, the interaction between the NMDA receptor and the PDZ domain, which we do now know, we have the structure of those, we can find regions that are specific for that interaction and basically begin to lock those down. So we can use other small molecular molecules to hold those interactions in place.